We are going to get started and we'll review the norms before we begin, which is to start and end on time, be respectful, and agree to disagree. The fellow being observed will have 5 minutes to set the context for the lesson and describe lessons being taught. States the age level and grade of students, states what part of the day this lesson took place, explains any unusual and important facts that occurred that day and may have impacted the lesson, explains what the lesson that was videotaped was on, explains what the lessons before and after this particular lesson was on, and explain how this lesson connects with the course of study. You ready, Jen?
Okay, so my students are in 7th grade. They are 12 to 14. So my classroom is a science lab, which has all the science materials for all the grades, so that's kind of a mess. There are 24 kids in the class. 7 of them have IEPs. 2 of those are full inclusion students, so they would have been in a sub-separate room, but now they're in that classroom. There are 11, this is like the ELL section, in 7th grade, so there are 11 students who are ELLs. Most of them are level 3, but there are a couple who are lower levels. In Boston, we use the FOSS curriculum. And this is the beginning of the Diversity of Life, which is their 7th grade life science unit. So we, the first thing that we talk about in that unit is understanding the characteristics of living things. So they've been learning these characteristics, and this was the final portion of that lesson. We've done very few claim evidence reasoning, so this is new to them. They've never used the results of an investigation for evidence before. They've done similar work in ELA, but they don't do exactly the same thing. So some of them wanted to use evidence only from the text. The lesson took place in the morning. It was a little bit unusual because it was the week before vacation. And so the days before the lesson, I was in charge of bike week, so every afternoon instead of having class I was biking with my kids. So that week they only had science class I think twice, and then the next day there was like a dance, so they weren't going to see me that day, so it was like one of 2 lessons the week before vacation, and I said, oh, I have to videotape and we have to do all of it today. So I tried to slam it all into less than an hour, and this is what we got. So
So for the next minute, the fellow being observed, I say Jen. Sorry. If you could restate the questions from the pre-observation forum that you had about your lesson.
So were they engaged? Is there another way that I could have gotten to the argument part faster because I didn't really have them engage in argument with each other? I was just wondering when you do tell them that yeast is alive. And then how can I make it more okay to not know all the answers? So, like some of them, I think you heard it on the video, they had this dialogue about like, we saw the yeast grow because we saw the balloon grow, but like when do you jump in and say like, no, that's not actually the yeast growing, that's just the balloon growing because of the gas. And a lot of them, I didn't copy the whole packets or bring them. But a lot of them, they had this checklist of like, did you see it grow? Did you see it reproduce? Did you see it, do this, do that? And a lot of them just checked yes for everything because they were making the claim like yeast is alive, so they knew it had to do all those things. So even if they didn't see it, they just checked yes. So how do you make it more okay for them to be okay not know it?
All right. So we already did the next section for the Form 3. So for the next 10 to 15 minutes, we will do the warm and cool feedback for Jen. And you can, for the facilitator, we'll make sure the conversation is focused. We will go around, we'll do one at a time again, and during this time you can not talk, you'll just take notes.
One of the questions you asked were the students engaged? So I have that during the introduction and directions, the students were off screen, but they were engaged, and my evidence for that was that they were asking appropriate questions. And then during the hands-on portion of the lab, they were actively engaged, putting together materials and taking notes.
I thought that you had really great activator questions. I liked how you were talking about what yeast is and, you know, it's associated with bread and, you know, and whether or not it's living or non-living, and the visuals on the board, and not only what, like, the picture they have in their worksheet, but also what it looked like under a microscope, which could have led the kids to think that it was living or non-living. They had a general understanding, kind of show that they knew what yeast was in general, which was really great, because sometimes kids don't really know what it is. They just think it's bread. It's not like or something that's in bread.
That's exactly what I was gonna say. I really liked how, you know, you didn't assume that, even though you covered it once before, some kids have a lot of prior knowledge and experience with different things, and other kids, it's just a totally new thing, and you were able to appreciate that and handle that really well, and answered their questions, and they felt super comfortable about saying I am, you know, just asking questions, which was great, because, you know, sometimes we do assume that, yeah, you've baked bread with your mom, or you've done things at home where you know, and not a lot of kids, you know, not all kids have those same kinds of experiences.
I liked how you gave them I didn't even know how to write this, the A-OK. I was trying to think of the word. It was maybe too late at night to say that they were not sure at this point. Like, I think sometimes that's really comforting to a student to say, you don't have to have a final answer yet. Even if after this you still aren't sure, you know, it's, so that kind of gave them the less anxiety going into it, so.
I like that there was a group discussion of the importance of documenting the results, even when the results are unexpected. And then you took that and you expanded it into the rationale for the double setup of both the control and the manipulated variables.
I was very impressed with their engagement, but the fact that you just gave them test tubes and all this stuff, and you just explained the steps to them, and they just went right to it. I think of my own classroom, and that happening would not look the same way. It would be spilled water and sugar all over the place. So you really demonstrated that you have really good classroom management, and also the kids are really used to that type of experimenting. So I was really impressed. I was really impressed with that, like with sugar and all that. I was like, no way I could do that with my kids. Really.
I liked how you had that reading available for them while they had a little downtime and they were waiting to record their results, and also along with that how you referred to the reading throughout the lab. So if they weren't observing something about yeast right now, they could refer back to the reading, and you had them use that throughout the lab, which I thought was great.
I think just to piggyback on that, I loved how the students used it. During the time where they were explaining it, there were so many students that said, and they referred right to that reading, even though they may not have understood why they had to read that while they were waiting, or maybe they thought it was a filler piece, but they actually, when they went to talk about their claims or their evidence, they went right to that reading. I read that, so it was, you could see them actually, it was a benefit, and they used it.
Another benefit that I thought was really good was the demonstration. Kids don't know how to add yeast and sugar to a test tube, and you know, you gave them the visual for that, and then they were able to go ahead and do it.
Student asked you a question at 35 minutes and 13 seconds. I don't know what the question was, but you know what, I liked your reply. You said, I don't know, what do you think? I thought it was just so great, so simple. It's so easy just to try to give them hints or to give them an answer. Something like middle schoolers are good at trying to give an answer. And you just cut them off and said, not in a good way, I don't know, what do you think? I was like, yeah, what do you think?
I really liked how you mentioned that there was a checklist to refer back to with the characteristics of living things, and I think it was totally fine that you didn't cover all those characteristics in one lab. There's just no way you could have done all that. So I thought that was great to you know, focus on one or two and to explore those, and I thought the checklist was just great to help them focus back on, like, why are we doing this, you know, and you kept repeating
the same thing. I think we're talking about different things, but I liked how you gave one of the groups the idea of checking off the directions after each step. Like they were struggling with did I do everything? And you were like, well, why don't one of you check off after you do each step to make sure? I thought that was a great idea. I'm like, I can use that all the time in my class. So just to make sure that was good.
Throughout the lesson, you gave them reminders. You said, okay, it's 10 minutes, it's 15. So you helped them with the timing to keep them on track for their observations. But you also incorporated in it, okay, you have this much more time to finish up the reading and the writing piece. That's good.
I like that you didn't tell them in that class period that use was alive. I really like that. I think that'd be something great to mention your next class when they're finished with their CERs. But I'm glad that you didn't tell them.
I liked how you incorporated like the whole data collection piece and how when kids had different results, like, well, their balloon, that balloon here, that's the same one in my desk and it's doing something totally different. How can that be? And science is messy and you're not going to always come up with the same results, and even though you're using the same materials, because what did you do differently that they did that, this may give you some kind of different results. So it really gave them a good feel for what science is all about. It doesn't come out perfectly every single time, and there's reasons why. So, and you had to think about that. You just didn't say, well, you probably did this. I like that. It's good hands-on science.
And I liked how there was a lot of built-in review kind of throughout. So just the little question of the balloons. So you asked what the balloons were for, if the students knew, and kind of reviewing that, or just a review of metabolism and why we were doing this. And it was just kind of throughout the lesson that there was a lot of built-in review and questioning to kind of get there activated and thinking.
I'm done. Everything I have has been said.
I just said I liked when the group got back together to discuss this whole and compare the differences between each group. So I thought they got really engaged with a little bit of that discussion back and forth. So.
I just wanted to add that I wrote down the questions that I liked how you said, what does this make you wonder about? What surprised you? I just love those questions. Sometimes we go just for like, what happened?
Yeah, no.
Like, what surprised you?
Okay, so if we're all ready, we can start the cool feedback.
Okay. You identified the question in the lab, is yeast alive? That was great scaffolding for your introductory lesson to CER. I'm wondering if the students needed a reminder at this point that from the question, is yeast alive, they could have made their claim that yeast is alive or yeast is not alive. And could they have done that at the beginning of the lesson before they actually started mixing the sugar and the yeast?
While I really like the activator a lot, perhaps using the short CER example or question as your activator or as part of your activator would have demonstrated to the students that observations from this experiment would be put into the CER format.
That's kind of what I was piggybacking up to, but you might have already done this, like, you might have already gone, it sounds like they've done some claims and evidence, so they probably already maybe knew that piece about it already, but I was thinking maybe if you had just kind of just posted on the board, the steps involved in that, or just a quick refresher, but then again, they might have that in their notebook, so that's just a suggestion if they didn't already do that.
I wondered, this is just more of a lab procedural thing that I was wondering, because they seem to all struggle with the initial yeast test tube water, some overflowing. But I wondered if you just have them mix it in a beaker first, and then pour it into 4 test tubes, or they could actually just stir it and do it slow, and it would just, and then pour it into 4. I just thought that might work better.
I'm wondering what the acronym green car stands for. I didn't hear anyone use it. It was mentioned in the pre-observation form. I know it's got to do with the characteristics, but my district uses Pearson, and I tried to match our green card to the characteristics we use, and it just didn't match, so. That's right, so maybe that should have been in the question instead of, yeah.
And you already mentioned this when you introduced your lesson, perhaps taking the lesson, spreading it out over 2 class periods. It felt like you had put a lot of work into one period, which you did, you already said. Perhaps break it up, you know, and integrate the whole CER process, like throughout the lab, and take little breaks maybe, and like do like little think, pair, shares, but you didn't have that time to do that. I understand that now. So, but I think that this could have been done in 2 periods. And then you could have really gone into the whole CER. And as far as the argument, that was one of your concerns. I think that if they're getting to the writing part, you can do the argument later. If they're still new to it, or if they're not new to it, then you can do the argument. But I feel like that's not necessarily something you have to jump into right away.
Mine was more of a question because I wanted to, you had asked, is it okay if they, you know, you were afraid that they just checked off all of yes, or, and this may have happened, but on your checklist, did you have, like a space for yes, no, and then, not sure, or still need to test, or something like that? But I felt you orally said it, like you gave them the, you know, you reaffirmed that it was okay, that they still were questioning. So I was surprised you said when you looked at the information that they checked on yes.
I was wondering, so 25 minutes is a long time. I'm thinking of my students, and to have them waiting in every 5 minutes to me. I'm wondering if there was something else with the reading to incorporate that, maybe pulling out evidence from the reading. I don't know if we had that, but maybe having a worksheet that went along, or something where they could actually pull evidence from that, and then that would help with the homework where they had to come in and then add the data, the actual hands-on evidence, and then create them. Any more?
Oh, I'm sorry, I don't have any more.
Okay. Hello.
Sorry.
So, same thing with the downtime. I agree. There was 25 minutes, and they were really well behaved.
Yeah.
Really, they were really well behaved. My kids not. I don't know about that 25 minutes, but maybe not only using the evidence from the reading, but also setting up their CER with a template or like a form like you had, and just kind of following along. And even they can even record data or as evidence while they're observing, because there's still some things happening. They can write a little bit. It's in pencil. They can always change it, and they could use that in addition to the writing that they get. So while they're doing all this, they're waiting for magic to happen. They're able to work on the CER part, or you can just like come and go around, check in on them, make sure they know what they're doing, or you can help them during that time.
Any other cool feedback? Oh, good, because we're at 14 minutes. We'll stop there.
Wow.
Good. All right. So now we have 5 minutes that we can ask clarifying questions about the lesson to Jennifer and whoever wants to begin.
Okay, I only had 2 questions, and one I already mentioned in terms
Do you
of.
want me to answer it now?
Yeah.
Yes.
The green car.
The green car.
So grows, reproduces, exchanges gas, eliminates waste, needs water, and then consumes nutrients, are made of cells, and responds to either
Okay.
the
Are
environment.
you writing that down?
I
Yeah.
wanted to.
Wait a minute.
Grows.
Repeat again.
Grow.
Growth.
Yeah, you reproduce this. So eliminates waste,
Eliminates waste.
exchanges gas, needs water,
Yep.
Processes.
we do consumes nutrients, and then are made of cells. I take the kids to Thompson Island for like a 3-day, 2-night thing in 7th grade. They go overnight,
Wow.
and they use that there. So I just started using it in the classroom, because then they're all like, "Oh yeah, green card. We learned this already. Like we did this."
Oh,
And
that's
they're
cute.
at school.
That's so
And they're-
great.
Yeah,